All right, good afternoon, everyone. My name is Parita Tisuwan. I am a medical doctor and a research fellow in Dr. Manstein's lab at Mass General Hospital. And today I will be talking about a novel 40 micrometer spot size fiber laser and how it compares to the CO2 laser for laser-assisted drug delivery. I have no disclosures to report. So briefly, the term laser-assisted drug delivery refers to the use of fractional lasers to bypass the stratum corneum, thereby enhancing drug uptake through the skin. And currently, the CO2 laser is one of the most commonly used devices for LADD. And in this study, we introduced a novel 40 micrometer spot size fiber laser, also known as the DFG system. Now, the DFG laser generates two different wavelengths, 3050 and 3200 nanometers. And it can produce a spot size as small as 40 microns, making it, to our knowledge, the ablative fractional laser with smallest spot size to date. Here's some histology that demonstrates the difference between the DFG laser on the left and the CO2 laser. Now, you can really see the difference in spot size as well as the coagulation zone thickness between these two lasers. So when it comes to drug delivery, coagulation zones, which is this area of thermal injury surrounding the ablation zone, plays a huge role. And this combination of small spot size and thin coagulation zone really presents the DFG laser as a promising candidate for LADD. So the goal of this study really is to compare drug uptake between the two lasers. Briefly on our methods, the experiment was conducted on ex vivo human abdominal skin. Laser exposure was done either with the DFG or the CO2 laser. And then immediately after, a fluorescent PLG nanoparticles were applied to the skin, and samples were analyzed using fluorescence microscopy. So our results showed that, first of all, both lasers were able to generate the same ablation depth. The DFG on the left generates much slimmer and more homogeneous channels at all depth, whereas the CO2 laser produces this cone-shaped lesions which narrows with increasing depth. Now, in terms of drug uptake, the nanoparticles remain trapped within the coagulation zones of both lasers, and this isn't entirely surprising because coag zones are known to absorb drugs initially before they diffuse into the surrounding tissue. So this panel shows horizontal sections taken at different skin depths, starting from the epidermis all the way down to the deep dermis. And again, it really highlights the difference in spot size as well as the coag zone thickness between the two lasers. So compared to the CO2 laser, you can see the DFG laser produces much smaller and more consistent spot size as well as coagulation zone at the same depth as the CO2 laser. This is also reflected in the fluorescence uptake, which you can see is much more homogeneous throughout the skin depth, whereas in case of the CO2 laser, most of the drug accumulates superficially and then decreases with increasing depth. Due to this, the DFG laser's small spot size, higher number of microchannels per area is possible at the same treatment density, which in our case, we use 1%. And for drug delivery, having more microchannels per treatment area significantly increases drug uptake and could also potentially minimize side effects. Looking at statistics, fluorescence intensity was quantified per single laser channel, as well as for the treatment area as a whole. Now, if you look on the left, you can see if we were to look at one single spot size, because the CO2 laser generates much larger spot size and coagulation zone, more of the test drug also accumulates within that larger area, leading to significantly higher fluorescence uptake superficially in the CO2 group. However, if you were to look at the total treatment area as a whole, you can see that the DFG laser can facilitate significantly higher drug uptake at all skin depth. Now, this is relevant because in clinic, it really is the total treatment area that determines treatment outcome rather than the single laser spot. And in this study, we show that the DFG laser can facilitate significantly higher fluorescence uptake compared to the CO2 laser at all skin depth. So to sum it all up, the DFG laser generates smaller and much more homogeneous channels and coagulation zones. This leads to significantly higher and more consistent drug uptake at all skin depth. This could also be associated with less side effects and faster wound healing, which is something that we plan to investigate in an upcoming clinical trial. Thank you very much.